So now you can see my presentation.
And please click on the bottom right. There is a start. Yeah, that's.
Yeah, it doesn't want to be clicked. Okay, that's it.
Okay,
so,
you can go on like this.
Okay.
Okay, it's no problem at all. I can see.
Okay, is it a full screen or you? No, it's not.
All right. It's all right. It's
okay. Sorry, but I have the full screen on my laptop. Sorry.
I see.
Okay, I'll just continue. So we have the retinal protonated Schiff base in both types of rhodopsins, but then the retinal in its old transform is found in microbial rhodopsins, while the 11-cis isomer is found in animal rhodopsins. But in both environments, we believe that the photoisomerization occurs on a very fast, ultra-fast sub-picosecond time scale, and the reaction is highly specific. Which means that only isomerization across a particular bond occurs. And also, as you already know, that the photoisomerization reaction proceeds with a very high quantum yield. But still the question is whether we understand how the protein affects the photoisomerization efficiency and time scale. And for that, we would like to know what is the intrinsic photoresponse of those chromophores in the gas phase. And to study the intrinsic photoresponse, we take out our chromophores outside the native protein environment, and we study their photodynamics and mechanisms of photoisomerization, both using theory and experiment. And since our conference, the title of our conference, Experiment Meets Theory, or vice versa, actually theory meets experiment, I will also pay some attention to the experimental details. So in the gas phase, we use what is called pump probe femtosecond action absorption spectroscopy technique. And this technique appeared just only recently. And this is because we are dealing here with the charged biological chromophores, and these are positively charged biological chromophores. And the density of these molecular ions in the gas phase, they are very low. And so we cannot use, ordinary simple registration techniques to be able to
sorry,
see.
Anastasia.
Sorry.
I'm sorry. Sorry to interrupt.
Yeah.
So the people not seeing your slide. So I think that you are sharing the, maybe you better share the PowerPoint. There are a couple of different ways of sharing. You can share the screen or you can share your PowerPoint. You have a couple of options. Take your time.
I'll try.
Yeah.
To share.
Share, you can PowerPoint or you can share the screen.
What about now?
The same one. Did you, maybe you can have another option when you share the screen.
Yeah, I'm trying to make two screens to put my demonstration on. Well, I mean, usually the way that I do now works. I don't know what's going on. So, but now you don't see my presentation. You don't see my slides.
I can see your first slide. So, oh yeah, I can see the second. Third. Yeah. This is, it works. It works this way. Now I can see third one.
Okay, this one. Can you see this now?
Yeah.
Everyone?
Yeah, third slide. Yeah, we can see. Yeah.
Okay, and now the fourth one, it works.
Fourth one, sorry. Fourth one, no. So I think that you could choose. Are you using two screen, right?
Before I had used only one, but now I'm using. I'm trying to share all my screens to all devices. Oh no, maybe not now. So what about extension?
Maybe you can try to. There is an option you can share your PowerPoint only rather than screen.
But I think this is what I'm doing actually.
Oh, okay.
I'm just sharing my PowerPoint. Then
Oh,
I'm
I
not
see.
sharing the screen.
Oh, I see. I see.
What about the screen? We can try to share the
Maybe
screen.
we can. Yeah. That's also. Yeah. This is much. Yeah. This is much better.
Yeah,
I
but
can see
now I
the-
cannot change the slides.
Cannot use. Yeah. It's changing now. Yeah. It's
Okay.
changing. Yeah. Perfectly.
Okay.
Yeah.
So
Perfect.
now
Perfect.
it's fine. Oh, I'm so sorry about that.
No problem. No problem.
Sorry. Okay. Coming back to the gas phase studies of the photodynamics, of the photo-induced dynamics of the retinals. So we use the experimental setup and we also use our theory. And I'm just feeling that I have to be maybe a little bit speed up, but nevertheless, okay, for the experimental studies, we use pump probe femtosecond action absorption spectroscopy technique. And this is done in the group of Professor Lars Andersen at Aarhus University, Denmark. And the problem is that to study the photo-induced dynamics in the gas phase, it is quite difficult. And this is because we have a very low density of molecular ions in the gas phase. And what we need to use action absorption spectroscopy technique, which means that we allow our molecular ions to respond to absorption of a photon. And for example, they can fragment after absorption of a photon. And this neutral counts we can detect, and then we can say whether the molecule absorbed the photon in the first place or they didn't absorb any photons. And using the electrostatic ion storage rings are very important here because we also get timing information. And basically we record the action on the time scale at which action takes place. This timing information is quite unique and can be used to separate out several excited state decay channels. And also this timing information is very important when we use the pump probe femtosecond action absorption spectroscopy. Why? And this is because we have somehow to have some means to differentiate between one photon or two photon absorption events. And since we cannot register absorption or simulated emission directly, then we have to be able to say whether our positively charged retinal molecule has absorbed one photon or two photons, to be able to at least to say whether, for example, the molecule is still in the excited state or not. And this timing information here in the electrostatic storage ion rings can be used for that, because when the molecules absorb two photons, they will decay faster. And this timing information will be used to differentiate the way whether the molecule absorbed only pump or probe signals or together, pump and probe. So that's, we end up basically in the 2D time resolved spectroscopy. Okay, and on the theoretical side, we construct and analyze potential energy surfaces, and we use quite high level of quantum chemistry to do this. And we do not do dynamics as Massimo, but as for now, we just only make this static pictures and study the mechanisms just by looking at the potential energy surfaces that are obtained with a very high level of theory. And we particularly are interested in using those theories that can provide a balanced description of both excited and ground states along the isomerization coordinate. That means that we might be able to describe and treat the excited states properly close to the Franck-Condon points and also in the vicinity of the conical intersections. So let's proceed to the outline. I will very briefly say which particular method we use from the quantum theory perspective. And this will be extended multi-configurational quasi-degenerate perturbation theory, which is very effectively implemented in the Firefly computational package. And then we will see on some of our results on photoisomerization and ground state isomerization of the retinal in the gas phase. So what about the quantum chemistry method? We use what is called an invariant approach to multi-state, multi-reference perturbation theories. And in particular, I think I don't really have time to go into details, but I will be happy to answer the questions if there are after my talk. I'll just only mention that we use state averaging KSSCF procedure to obtain our zero order vectors that span our model space. And since we use not the entire KSCA Hamiltonian, but only a few states, then we work with partial contraction in our primary space. Then we use one particle fork-like zero order Hamiltonian, like for example in KSPT2 theory, which is quite similar in this regard. And there is only one obstacle or one interesting question on how to define this H0 Hamiltonian, which is written here. And this H0 Hamiltonian is actually defined differently in the MCQ-DPT and X-MCQ-DPT theories. And the MCQ-DPT without X is basically a non-invariant theory, while the extended version becomes invariant with respect to the rotation of the KSSCF vectors inside the model space. And this is because we use the entire H0 Hamiltonian, which looks non-diagonal in the basis of the KSSCF vectors. Okay, and then the zero order states that are allowed to interact with our KSSCF states, these are all possible configurational state functions which do not belong to our KSCI space. And these are obtained, of course, by single and double excitations. We have no construction in our secondary space, and that's the difference between, say, MCQ-DPT and KSPT2 theories. Okay. We actually think that the invariance of X-MCQ-DPT, which the theory itself has been developed and implemented into the Firefly package by Alex Granovsky. And we think that the invariance is quite important when we really want to study very carefully the topography around the conical intersections, because the invariance is crucial when we are dealing with the KSSCF functions which share a common set of leading configurational state functions or Slater determinants. Okay, hopefully you believe that the X-MCQ-DPT should be a nice, quite high-level theory, really to treat the conical intersections in this case. And this is not the only theory that can be used, of course, here, but this is our lovely theory that we always use because it is very efficiently implemented. Okay, now we proceed to our results, and then first we look on the photo-isomerization mechanism. So with by using this X-MCQ-DPT theory, we construct and analyze one-dimensional relaxed potential energy scans in the first excited state. And what you can see here, these are the all-trans chromophore and 11-cis chromophore. First, let's start with the all-trans chromophore. So the all-trans chromophore, it has barriers in the excited state, and these barriers, they hinder intramolecular rotation. Moreover, we can make the scans and analyze them for different double bonds here. And what we can see is that the barrier is smallest for the rotation across the central double bond, which is basically 2 times smaller than for all other bonds. And this tells us that there is a specific isomerization going on for the all-trans chromophore in the gas phase, but the bond, this specific bond is different from that in the protein environment. Okay, and for the 11-cis chromophores, the potential energy scans looks like that there is no any barrier. And this is indeed the case. There are no any barriers here in the excited state, or at least no barriers at this level of theory that is written on the slide. And now what we would like to do, we would like not only to say that there is a barrier or there is no barrier, but we also would like to make estimations for the excited state lifetimes to be able to directly compare to what we measure or to what Lars measures experimentally for the retinal in the gas phase. And for that, to estimate the excited state lifetimes, we use a statistical theory. We use a transition state theory, and sorry, the equation is kind of here. This is a slightly modified version, but nevertheless, what we need to know the activation barrier, and we also calculate the statistical prefactor in the direct way. And from this statistical prefactor, we basically use the partition function that is related to our reaction coordinate. And since the potential for the reaction coordinate, this is the potential that hinders intramolecular rotation in the first excited state, is highly anharmonic, then we solve the one-dimensional Schrödinger equation numerically to find the eigenstates, to find the torsional levels that fit into this potential. And these states then are used to calculate the partition function. Within the help of this transition state theory, we can estimate the rate constant, and we can also estimate the time scale as the reciprocal of this k. We have estimated the rate constant and time scales at 2 temperatures, and also the experiment was also carried out at 2 temperatures. And as you can see, this statistical theory works actually quite well. And more importantly, we can indeed predict the time scale, for example, at 100K, which is almost 100 picoseconds. And at room temperature, the excited state lifetimes is also quite long. It's picosecond, like 4 picoseconds on a picosecond time scale. And more or less the same is found when analyzing the experimental dynamics. So what is the message? The message is that even in the gas phase, without any environments, and especially without any solvents that can influence the dynamics quite a lot, we see that the time scale is 1 order of magnitude longer in the gas phase for the all-trans chromophore compared to what we observe inside the protein. We have also calculated the photo-induced absorption. This is basically to answer the question whether the S2 state can interfere. The S2 state basically does not interfere really in the gas phase to the reaction coordinate, and there is no like huge mixing between S1 and S2. We can also say that the brightest transition when we go along the reaction coordinate is S1, S3 transition. And it undergoes a blue shift. And this exactly the blue shift that is observed in the experimental studies. So indeed, we see that at first there is an absorption around 700 nanometers that gradually goes to 660 and then 620. This is already the absorption from the hot ground state. So these results are also in good agreement, our results in good agreement with what we find experimentally. And to make conclusions to this part of my talk, I can say that what we have found or what we have learned from statistical theories, that the all-trans chromophore is intrinsically much slower than the 11th cis chromophore. 11th cis chromophore that was found experimentally has an excited state lifetime of 0.4 picoseconds, which is quite similar to what is observed inside the rhodopsins. But the all-trans is really like 1 order of magnitude slower. And moreover, the protein not only changing the time scale, it only changes the specificity of this reaction. Now we ask ourselves, can we somehow tailor the photo-induced dynamics? Can we somehow change the time scales and specificity? And for that, we use, of course, the chemical modifications, as many would think. And we introduced 2 kinds of modifications to our all-trans retinal. We first just change the termination, which shouldn't actually affect much the time scales. And we would like also to lock the lowest energy pathway for the all-trans isomer. Because by locking the lowest energy pathway, one would think that the time scale will be longer and the lifetimes will also be longer. And now we will see what happens in reality. So we will compare what we have in our modified retinols compared to what it was for the normal all-trans retinol. So as you can see here, this is the red curve. This is the potential for intramolecular rotation about the central bond in the normal retinol. And now we switch to the demethylated. And as I already told you before, I think the dynamics shouldn't be really influenced by the modification of this tail. And indeed, if we just calculate the potential that hinders intramolecular rotation, the potential looks pretty much the same as it was before. But experimentally, what we see, even the time scales, the lifetimes are quite close to each other. Since generally we see that the time scale for the demethylated Schiff base is reduced compared to the normal retinol. And why is that? This is actually the purely kinematic effect, because in the gas phase, we have different reduced rotational constants, and the rotational constant for the demethylated version is 2 times larger compared to that in the normal retinol, and this leads really to the reduction of the lifetime. But then let's move to more interesting example where we lock our C11, C12 double bond. Okay. And unexpectedly, we see that the potential that hinders intramolecular rotation now about the other bond, which is the C9, C10, which is depicted here, is very different from what we expected beforehand. We expected something like this blue line that was observed in the normal retinol with quite a large barrier that hinders intramolecular rotation. And now the potential looks highly asymmetric, and also the barrier from one side drops to basically nothing. And of course, by looking at the calculations and by looking at the structures, it is very easy to see that the structure becomes non-planar also in the ground state and in the excited state. So here you can see the structure in the excited state, and it is highly twisted. So that, of course, explains why the barrier drops dramatically when going to this locked version. Interestingly, okay, we can of course calculate the time scales and compare to the experiment, and again we see that our statistical predictions are quite in a good agreement with what we find experimentally. Interestingly, we have the time scale for this photoisomerization, which is now very close to what we find in the bacterial rhodopsin. So it seems that it's very easy to tailor photophysical properties and photochemical properties of the retinol by making smart modifications. Another thing which is also quite remarkable, that this particular locked chromophore can undergo the unidirectional rotation, and it's not only from all trans to 9 cis as depicted here, but also when you go from 9 cis to all trans, it completes the cycle and the rotation proceeds in the same direction. And what we can say that this locked version can indeed be quite promising for designing red light driven molecular rotary motors, because first of all, it's driven by red light. The second thing is that the full rotational cycle goes here only in 2 steps, and these steps are ultra fast and photo-induced. So there is one absorption of 600 nanometer photon from the all trans going to 9 cis, and then from 9 cis another step going to the all trans again. And there are no intermediate states in the ground state which possibly could slower the entire cycle. So this is quite nice, and I think this idea can further be somehow developed. Okay, and now just a few words about the ground state isomerization. And why we are so interested in the ground state isomerization, because the ground state isomerization is here coupled to photo-induced isomerization, because we have this conical intersection, which is located very close to the saddle point in the ground state. And the presence of the conical intersection also leads to bifurcation in the ground state. And this, many, I think, of us know this, but this is just a very nice demonstration of this. So we have this pathway going from one isomer to the other, and we also have this pathway. From the theoretical perspective, the presence of this conical intersection might be a challenge, because we need to take into account that there is this conical intersection, first of all, in the first place. But then there is another thing behind this story, and the story is, what is the activation energy for the ground state isomerization? And the story behind this question is comes from also experimental studies. Quite recent experimental studies that were performed, I think, in 2015, they found, the authors of the studies, they found that the ground state isomerization barrier is much, much lower in the gas phase compared to what is found and observed in the protein environments. And it might well be the case, because we know that the environment can influence the isomerization. So why not for the ground state isomerization? But then, on the other hand, from the theoretical point of view, we would like to reproduce this value in our calculations. And so we do this. We try to locate the barrier in the ground state, again, using our XMCQ-DPT theory, and we follow the next steps, the following steps to find the transition states. So first we locate our conical intersection, and then we perform an unrelaxed 2D scan in the branching plane of this conical intersection. And we find the starting point for our optimization of transition states in the ground state. So basically it's somewhere here and it's somewhere here. The transition states are then located using the XMCQ-DPT theory, and all points and everything, they are located using this high level of theory. So here you can see the detailed view of this part of the potential energy surfaces in the ground state, which is close to the conical intersection. Okay, so what we have found? We have found that the barriers are quite high. They are more or less as high as they were observed inside the protein environment, something like 1.2 EV. And we also calculated the rate constants, and the rate constants are also in a quite good agreement for the red absorbing proteins with what was found experimentally. So what's wrong? What's wrong either with our calculations or with the experiment? And since I kind of don't really have much time to discuss the experiment here, I'll just only mention how we proceeded to somehow correct the experimental results. Of course, these experiments are also quite complex, and these experiments, they are based on this multidimensional ion mobility spectrometry with the collision-induced dissociation combined with mass spectrometry for registration of the products. And these experiments, they are performed in the gas phase. So molecules, they are travel in the tube filled with some gas. And at some part of this tube, the molecules are activated by applying some voltage. So by applying a high voltage, then more energy is transferred through collisions, and the reaction then eventually will happen. So in the experiment, they look at the threshold voltage when the reaction actually starts. And this voltage, the experimentally applied voltage, it is divided by the number of vibrational degrees of freedom in the molecule. And this voltage is correlated to the activation energy. This correlation is purely empirical, as it was used in the experimental studies, and it was obtained by analyzing some reactions for a number of molecules with not so high number of vibrational degrees of freedom. So as you can see here, so they did this for, say, 10 compounds, and then they see that there is this linear correlation. And just to be sure that you understand what we are dealing with, here I just also put the mark where the retinal should be. The retinal isomerization in the ground state should be. So that's the green point, and this green point is obtained or put on this line where we have actually to extrapolate the experimental points to this lower part in the graph. Okay, it is obviously that the retinal has a lot of degrees of freedom, vibrational degrees of freedom. It's almost 200 vibrational degrees of freedom. And the question is then, is there any theoretical basis under this empirical correlation? Can we somehow provide this? And we do this, we introduce a theoretical model, which is based on the quasi-equilibrium theory. And this in quasi-equilibrium theory, it is applied to the microcanonical, to the systems of microcanonical ensemble. And with the help of this theory, we are able to calculate the rate constants depending on the total internal energy that the molecule has. If we fix our rate constant, which is basically done in their experiments, we can say that the activation energy can be correlated with the total internal energy. But to get the analytical expression for the activation energy as a function of the total energy, we need to make some assumptions. So if we do them, for example, we go to the classical limit, we also assume that the energy that the molecule have, they are much larger than the activation energies. We can, in principle, end up with some kind of the linear correlation between the activation energy and between the total energy divided by the number of vibrational degrees of freedom. But the problem here is that we also have a prefactor. And this prefactor tells us that basically we should be dealing not with the total number of vibrational degrees of freedom, but only with those that are active upon certain conditions. And this effective number of vibrational degrees of freedom, they are they are dependent on this n, and the different curves will be obtained for different n. And moreover, this effective number of vibrational degrees of freedom is also a function of the total energy. So we might expect to have only a linear function in a limit where we have, where this prefactor is basically close to unity. So if we do it correctly now, based on our theoretical basis, based on our calculations, then we can show that the curves that are used to correlate the voltage and the activation energy, these curves, they are different for the molecules of different size, and the shift is quite large for the retinal. And then the next thing is that we have a bend in the very low area of this correlation curve. So when we use the curve calculated by us, we actually get the experimental energy barrier for the ground state isomerization, which is close to 1.1 eV, and this is very close to our theoretical predictions. So this red curve here is this empirical linear dependence, which at this particular voltage or that particular voltage gave us only 0.6 eV, and with the corrected curve, we get the activation energy of 1.1 eV. Okay, so I think that was also a very nice example where the theory and the experiment can work together. And now I just switch to conclusions, and I hope I have demonstrated that theory and experiment can really work together, and it's quite important that we have experimental results that can be directly compared to the to our theoretical predictions, and also we can use our theory sometimes to interpret in a correct way the experimental results. What I have talked about during today, I also like enumerated here. So we have studied the photo-induced dynamics of the isolated retinal molecules, and these dynamics can be significantly accelerated and also steered. We can also get an ultra-fast rotary molecular motor. And finally, we redefined the experimental value for the ground state isomerization and show that the barrier for the ground state isomerization is as high as in the protein environment. Okay, and I would like to thank my group at the Moscow State University, and of course I would like to thank the experimental group of Professor Lars Andersson. And from my group, I would like to thank Adil Kabalda and Pavel Kusochik, who really contributed to the studies that I talked today. And thank you very much for your attention, and sorry for technical difficulties during my talk. Yeah.
Okay, I think the one quick question, please. Okay, so oh yeah, okay. Okay, Anna, go ahead.
I'm just curious about your method